good morning, hola, bonjour, guten tag, ciao, namaste, ni hao, aloha, konnichiwa. It's our pleasure to welcome you again to another series of global IMMUNOTALKs where we hope to create a world community focused on the renowned research in the field of immunology and microbial pathogenesis. And it is a great pleasure today to have our speaker, Dr. Lalita Ramakrishnan, or Lali, as we like as she likes to be referred to. Lali is a professor of immunology and infectious disease. She's the head of the molecular immunity unit at the University of Cambridge. Lali started her PhD at Tufts University in Boston, where she was a graduate student in Naomi Rosenberg's lab. She went on to do her postdoc fellow at Stanford with Stanley Falco and did some phenomenal work. And she's also, and then went on to be an assistant professor at University of Washington before moving to the University of Cambridge, where she is now. She's also, though, a physician, and she has been working in the area of infectious disease and tuberculosis, and has been, again, a world-leading authority in the pathogenesis of tuberculosis, this devastating disease, and also has made fundamental new insights into how the Mycobacterium interacts with the host and host cells, and how the host cells respond to inflammatory events that can lead to containment of the disease, but also pathogenesis of tuberculosis. Lali has had many awards over the years, to name a few, starting back from when she was first starting her career. She's received a Burroughs Wellcome Pathogenesis Award of Infectious Disease. She's received NIH Director Pioneers Award, Merit Awards. She's a Wellcome Trust Principal Research Fellow, and she was recently inducted into the National Academy of Sciences, as well as the Academy of Medical Sciences. She has served on numerous advisory boards for infectious disease and tuberculosis, and is a great pleasure to have her here. Lali, we'd like to have you ask you a question. As you know, we like to have our trainees learn from us and from your path and your career. And so you go ahead, you can turn on your screen if you'd like, your video, go ahead. Sorry, I didn't realize it wasn't on earlier. We were wondering if you could share, so welcome. We were wondering if you could share one of the most impactful decisions in your scientific career. That's a loaded question, so unload it.
I would say that it was the choice of postdoc and going to postdoc with Stanley Falco, and to take on this what then seemed a kind of wacky way to study TB. And the reason I think it was especially so was that wasn't why I had headed west from Boston to do. I was going to go to Don Ghanem's lab. They're very, very fine scientists and work on viral pathogenesis, particularly at that time hepatitis B. And when I got there, I was an infectious diseases fellow, and I was going to do this in conjunction with that. And I suddenly realized that the questions of hepatitis B were more solved than a field that I wanted to get into. And so I asked Don if I could work on TB, and he said, absolutely not. You're going to have to find a different lab. And he said, why don't you? So this is, and Falco didn't work on TB either, but, we then worked out a deal where I would do this. And as you'll see, that the idea to use a surrogate pathogen to study TB and so on, took off beyond our hopes and expectations. So I would say that is far and away the most impactful decision.
Great. And yes, and it's been a very impactful for the field as well. So we really look forward to hearing about your research. If you want to go ahead and share your slides.
So at the moment, I'm not okay. No. What are you seeing?
Your slide. Yep, perfect. Looks great.
Okay, good. Excellent.
All right.
And so everyone you can see me, you can hear me, and you can see my slides.
Yeah.
Okay. So I'm going to tell you today about how we've used the zebrafish model and a natural pathogen in it that is a close relative of TB to understand TB pathogenesis. And I'm going to specifically focus on the question of how TNF, that is one of the most recognized host resistance factors in TB, can cause pathogenic cell death that actually paradoxically leads to hypersusceptibility. And how this work that we've done in the fish is relevant to humans as well. Okay, so you've been invited to ask me questions on Twitter, but I've also given you my emails. Should you want to approach me that way, I'll be very happy to take any questions through that route as well. So TB is an ancient disease. You know, depending on who you ask, it's several thousand years old. This is a skeleton of a spine that was undoubtedly bent by a well-known disease of a well-known site of TB disease, the spine, and it's called Pott's disease of the spine. And incidentally, my mother had this when I was a child, and had several recurrences until what is now current day treatment came into play, and she was able to finally get cured, but she fortunately did not reach this stage. So TB, partly because TB is so old, it is numerically TB's humanity's greatest infectious killer. And this graph shows you how many diseases that we consider to be these great killers, for example, plague or smallpox, pale before the number of deaths that have been caused by TB. And if you were to look at COVID, terrible as it is because it's relatively new, it would be just a teeny tiny dot in the middle there. So, but, and this is historical, but TB remains a major killer. It kills anywhere in the range of 1.5 to 2 million people a year. Until COVID came along, it was, had the notoriety of being the greatest infectious killer still. And not only is, TB prevalent and a major burden in much of the world, as you'll see in that top graph, but also is multi-drug resistant TB is spreading, which and can get extensively drug resistant, that can convert TB to a almost incurable disease or very poorly curable disease. And there are at least 500,000 cases of this per year, and the mortality from this is much higher. So, but the paradox of all of this is, and this will not surprise you in the era of COVID, is that most people who get TB actually self-clear. About 90 to 95% of people will clear TB within months to 2 years. And this is work that I've done over the last few years with clinical and epidemiological colleagues, Marcel Baer and Paul Edelstein. So it's sort of my night gig. And we've come to this, and there's very fine studies from about 200 years even that show that most people clear TB. So that raises the question then, if this disease is killing 2 million people, but most people clear TB, then this 2 million people so 2 million people die from it, but 10 million people per year get TB. So that 10 million people represents about 5%. And so the real question is, why do these 5% that comprise an enormous burden get TB? What's wrong with them? And, if you look for, very profound immunocompromise, that's just the tip of the iceberg. Most people who get TB don't have any real immunocompromise. HIV is a major risk factor, of course, but the majority of people, and it is a substantial number of people who get TB have HIV, but actually most people who get it, the majority do not have HIV. So it begs the question, what's wrong with these people, so to speak, immunologically, what's wrong with them? And it's been clear, it's not surprising that as with any disease, infectious or non-infectious, that genetic susceptibilities influence TB outcomes. And this was made crystal clear by an unfortunate natural experiment that got done in 1929. So there's a vaccine for TB. It's a live attenuated vaccine. It's called BCG. BCG is not terribly effective, as reflected by the fact that we have lots of TB around, despite the fact that BCG has been given for almost 100 years. It does have it provides some protection to kids against disseminated TB, but then fails to give protection subsequently to those same people. But back in the day, when people were inoculating kids, they were just giving it to kids orally. In the north of Germany, which is genetically considered sort of homogeneous, somebody accidentally grew back, everybody grew their own BCG back then, and a physician there accidentally grew virulent TB instead of BCG and gave it to 250 newborns. And the interesting thing that happened was that they divided into a third. A third died of TB, these newborns. A third got TB but did not die. So they managed to eventually get rid of it. And a third survived without really very much evidence of disease. So this clearly showed that kids who got exactly the same dose, 2 doses, one at birth and one at 1 month, both all did very differently from TB, highlighting in a tragic way that there's differential host susceptibility. This is not too surprising if you consider that TB is a complex disease, like many diseases. Can you see my arrow, everyone?
Yeah. Mm-hmm.
Okay. So the first thing that happens is that the infected individual coughs up the bacteria, and the aerosols of these bacteria reach the unfortunate individual near them. They get gobbled up by alveolar macrophages, which then go deep, and now they start to recruit more macrophages to form a granuloma. And this is a hallmark pathological or immunological structure of TB, which consists of these highly differentiated macrophages, and then many other types of immune cells come in, and then eventually the bacteria can either get cleared or they can continue to grow, and eventually if they don't get cleared, they can cause necrosis of these macrophages. And this is a key pathogenic event because the bacteria can grow like gangbusters in this necrotic material. And not only that, this is all the necrotic granuloma is what causes spread, allows the bacteria to spread better than the cellular granule. So you can see there are many possible times when the bacteria can be cleared, potentially even before the macrophage. They can be cleared when they get into the alveolar macrophage. They can be cleared by the structure that forms if they fail to be cleared by the alveolar macrophage. And then even at this stage, they can be cleared, even though they become extracellular, although the odds become less and less. Okay. Now, when I became a postdoc, Stanley encouraged me to look for surrogate pathogens that I could use instead of TB, which grows very slowly, needs BSL-3 facilities, which he didn't have. And so I identified a pathogen that causes a skin disease, a skin and soft tissue disease in humans. And I managed to prove that for myself. Always the experimentalist and empiricist. And this pathogen turns out to be the most closely related to the human TB bacterium, as I found many years later when I got it sequenced. But I had many hints even back then that that was the case, and that my hunch proved correct. But it turns out that the pathogen also infects fish, and fish were dying. And this was identified by a guy called Aronson, because fish were dying in the Philadelphia aquarium that's now defunct. And initially they could see that the fish had the same tubercles that I showed you in cartoon form that looked like TB. They could even see that they had the characteristic bacteria in there. And yet they couldn't grow them until he had this idea that because fish were cold-blooded, maybe you should try culturing at low temperature. And lo and behold, the fish tissues did grow a bacterium that he named Mycobacterium marinum. And now it's going to be clear to you why you get disease that's peripheral in warm-blooded people like me, because the pathogen is adapted to low temperatures. And so in fish, it gives a systemic TB-like disease that involves, all these different organs. And but in humans, it gives soft skin and soft tissue infection that can get severe, but not if it's caught in time. So enter the zebrafish. So because we knew that it could give fish, all kinds of infection, we decided to try to use the zebrafish. And actually, this idea was given to me by one of my infectious diseases attendings at UCSF, a sort of polymath person called Don Payan. And he said, why don't you try the zebrafish and see if you can explore the host site? So, eventually we did. And the zebrafish gets TB. And this is why zebrafish with Mycobacterium marinum. So here's a human granuloma. You can see it. I haven't stained the bacteria. We haven't, the bacteria are not stained here. This is an H&E that stain that I got. But you can see that you've got an intact granuloma. You've got this acellular region in the middle, because that's where the bacteria have lysed the cells. There's no blue nuclei. But otherwise, you can see a lot of nuclei in there. In this fish granuloma, which I stained with a bacterial stain, you can see that same structure, cellular, but then in the middle is where the bacteria have lysed the cells, they've necrosed the macrophages, and they are really growing like crazy, just as I told you. In contrast, you can see a cellular region here where there's bacteria, but there are many fewer. But the really great thing about the zebrafish, which is why it's a pet of developmental biologists, is that they're see-through for the first 2 or 3 weeks of life. And so we wanted to know if we could see the TB granulomas in them, and indeed we could. And what you can see here is we've taken bugs that are fluorescently labeled with, GFP or RFP or whatever fluorescent protein you want. And you can see that granulomas have formed, and you can see there are dots which represent individual infected macrophages. And you can also genetically engineer fish to have green fluorescent macrophages. They have red fluorescent neutrophils in this case, and you can see a nice tight granuloma is formed where there are both infected and uninfected macrophages. The bacteria are blue, and you can see an occasional neutrophil, which is consistent with the human TB granuloma. But I just want to give you a feeling for how to work with these, because these larvae are tiny. And so if you want to get consistent infection, just like with, say, mice, you might infect the mice with measured doses. And so we micro-inject the fish with a hand-pulled glass needle. And we've got a vacuum tube on the right-hand side that you use to grab the embryo, and you insert the needle in there, and you get your infection. And that was the hindbrain ventricle cavity, but you can also use a little hockey stick to deliver the bugs straight into the vein of the fish. And this can be done at a pretty good pace. People can do it as at 100 fish per hour. And then because they're tiny, they'll fit into 96 well plates, and you can put them in these plates. And because they've got this great big yolk, they don't need to be fed for 14 days. So you can put your drugs in, you can do whatever you want. And then you can take that plate, put it on ice for 10 minutes, and that makes them stop swimming. They get a thermal anesthesia. And then you can put them on a stage like this, and you can do a rapid imaging, which will at least tell you how many bacteria there are, and you can quantify this. You can also do much more detailed imaging, as you're going to see in a bit. So with this all lined up, David Tobin came to my lab as a postdoc with the idea to do a forward genetic screen. And this brings me to the second great feature of the fish. You can do forward genetics in the fish relatively easily. And so you can mutagenize a male, and then you can cross him to females, and then you can take the offspring and do basically Mendelian crosses and identify whatever trait it is you're looking for. Developmental people look for developmental traits: no heart, no tail, funny eyes. We wanted to see if we could see infection that was different. And indeed, we saw that there were many, many different clutches of fish that clearly reflected hypersusceptibility. So here you've got a wild type, and you can see the nice granulomas, and you can see this mutant has the sibling mutant, so it's very powerful, but you're always comparing sibs to each other. You can see that it's got infection that's much, much more rapid. Now, what David showed, and so the rest of the story, we have many, many such mutants. Some we've published, and many that are ongoing quests in the lab. But I'm going to tell you the story of just one mutant and the rich dividends we've had from it. So what David identified was that this mutant had accelerated necrosis of the granuloma. And remember, I had told you that granuloma necrosis here is a pathogenic event. Well, that is what was going on. So here's the cellular granuloma. And I've shown this to you before. This is just to remind you. And his mutants initially looked like the cellular granuloma, but very rapidly looked like this. You can see the green macrophages are already going away. The blue bacteria are growing. And in just a few more days, you can see that the bacteria are growing, very, very prominently in these cords, which is classical for Mycobacteria growing extracellularly, and the cellularity of the granuloma is almost gone. So when David mapped the mutant, he found that it mapped to the arachidonic acid pathway. So these are 20 carbon lipids, they're membrane lipids. And what he showed was that it mapped to an enzyme called Leukotriene A4 hydrolase, or LTA4H, which catalyzes the conversion of Leukotriene A4, which is an unstable epoxide through a hydration reaction, to a highly stable and highly pro-inflammatory molecule called leukotriene B4. However, it turned out that the reason that this particular fish was susceptible was not because it had too little LTB4. Turns out that while LTB4 is highly necessary for many things, many infections, you don't need it for TB. There are other mechanisms. But rather, the high amount of the absence of the enzyme was pushing the leukotriene A4 hydrolase to make an anti-inflammatory molecule called lipoxin. And this was why the fish couldn't mount a good immune response. So to summarize then, what we had was a mutant that had low LTA4H. And we thought, okay, and this was giving us uncontrolled mycobacterial growth. But before we went too far to figure out why this was and what was going on, we decided to go to human cohorts. And so we found we identified a human variant in the LTA4H, a common human variant that was a C to T transition that was that caused, it was a causal mutation that changed LTA4H levels even in normals. And we went to a TB cohort and looked for severity. And the surprising thing was that the low LTA4H, that was the CC, had more severe TB. So that was not the surprise. But the surprise was that the highs also had more severity, and it was the heterozygotes that were protected. So this was a moment of slight panic, because the human was raising a question that we had not come across in the fish. So we went back to the fish. David went back to the fish, and he now created an LTA4H high fish. And to our relief, these fish also had uncontrolled bacterial growth and pretty much looked like the lows. So the fish looked precisely like the human. And then the final thing that we did at this stage was to show that the low LTA4H, because of that high lipoxin that came on as in response to the mutation, these fish didn't mount an inflammatory response. And in particular, we showed that it was the lack of TNF that caused this uncontrolled mycobacterial growth. And why that is the case is the subject of another story, another day. But what we found was that the high LTA4H made too much TNF, and it was that high TNF that was responsible for their susceptibility. So now let me just tell you a little bit more about the human cohort. So our mutation is here. It's 12 bases upstream of the transcriptional start site. And as I told you before, this is in normals. You can see that the CCs make the least protein, the TTs make the most, the CTs are in the middle. So it's a causal mutation. Our cohort was a TB meningitis cohort. Now, TB meningitis is the most severe form of TB. Carries a 25 to 40% mortality, even if it's drug sensitive with full treatment. And the reason is, you can see it's a basilar meningitis with a very severe inflammation that forms at the base of the brain in this unfortunate individual who clearly didn't survive. And even people who do survive often have very bad sequelae, neurological sequelae. Now, because of that sort of gemisch, that inflammatory debris, that mass that you saw at the base of the brain, people have thought about using an anti-inflammatory in conjunction with anti-TB treatment to see if they could lessen mortality. And this was being done physician determined all over the world, until Guy Thwaites, who's now become our close collaborator, decided to do a randomized controlled trial in Vietnam, and basically showed definitively that dexamethasone, which is a corticosteroid and potent anti-inflammatory agent, did decrease mortality. However, as you can see, the difference is quite small. And when we got this result that maybe people were dying from TB because they either had too little or too much inflammation, we wondered if the dexamethasone, the anti-inflammatory, was only helping those with too much inflammation. And when we, fortunately, Guy had saved blood and we could make DNA. And when we looked, we were delighted to find that was exactly the case. In the placebos, the highs are doing badly, and they're not dying at all when they're treated with dexamethasone. This is quite a profound effect. The lows might be doing a little bit worse, but might not be. And the heterozygotes are doing about the same. So all of this benefit was accruing from this one genotype only. So just to summarize this little bit here, we started with the fish, which come from the Ganga River, the Ganges in India. And then we went to Vietnam and showed this variant that we found in this genetic determined that we had found in the fish to be operant in TB meningitis in Vietnam. And then we also showed in a leprosy cohort the same disposition where the heterozygotes got less severe paucibacillary leprosy, whereas both homozygotes, the high and the low, got multibacillary leprosy. And this was done in a cohort in Nepal. So we then pushed on a little bit, and we, being Laura Whitworth, who's a human geneticist who's works in my lab and is my colleague, and what she did was to join up with some mathematicians here at Cambridge, Roger Sewell and one of his mathmo students, as they call him here, to do a Bayesian analysis now of a fresh cohort in Vietnam and a cohort in Indonesia. And Bayesian analyses are great for when you have small numbers of patients, and when you have multivariate analyses, as is always the case if you're looking at something over the course of time. And again, she was able to confirm that in Vietnam, the TTs don't die and the non-TTs die if they're steroid treated. But remember, because of Guy Thwaites' small benefit, now everyone's given steroids. So the only thing, we can't do the same trial, the same question, we can't do the trial again. We can just ask, do TTs die less than non-TTs? And indeed that was the case. And the same thing was happening in Indonesia, although the effect was a little bit less just because people weren't getting quite as good care because it wasn't a referral, it wasn't the kind of trial center that Vietnam had. And so these people were just dying more right from the beginning. Okay, now, remember, so now we had the impetus to really go back to the fish and see, okay, what is going on? Why is it that high TNF, high LTA4H through high TNF causes pathogenesis? Why isn't it just protective or even more protective than low TNF? What's going on? And this question was addressed by Francisco Roca, who came to my lab, and like David, he's also left now to start his own lab in Spain. David's gone to Duke University, where he's a professor. And now, what I told you before was that the low LTA4H doesn't make enough TNF, and what this does is that the macrophages don't control the bacterial growth very well, and then they die. But in the high, what we saw was that actually high TNF was controlling bacterial growth even better than the than the medium, the optimal. And yet these macrophages that had hardly any bacteria in them were dying. Before we went further with this though, we again went back to our human cohort. And this time in this new cohort, Guy had saved cerebrospinal fluid. And so we were able to measure cytokine levels, which were done with his colleagues there. And again, analyzed with Laura and Roger and new math students coming in. And what we found was that TNF in humans was indeed a pathogenic susceptibility factor. So you got, it'll seem paradoxically reversed, but remember, these are all steroid treated. So if TNF is a pathogenic factor that is corrected by steroids, then survivors will have high TNF levels. But this would mean that in the absence of steroids, which would be the normal way you would do the association, these people would die more. And so we see that the survivors are dying, you have to think of it as they're dying more if they have high TNF, or high TNF people are dying more. And this was the case if they had the TT genotype, but importantly, it was also the case if you removed the TT genotype and looked only at those who didn't have the TT genotype. What this was telling us was that excess TNF is pathogenic, independent of the height of the TT genotype. So the TT genotype definitely gives you high TNF and can cause death, but other routes to TNF dysregulation, you can imagine all kinds of them, also give you this, and it told us that TNF was a broadly acting pathogenic determinant and made us even more eager to identify how it was working. So when Fran looked, what he showed was that TNF was causing the infected macrophage to overproduce mitochondrial reactive oxygen species, which I'm going to call ROS from now on. And this was, these reactive oxygen species would initially diffuse into the lysosome and start to kill the bacteria, which is why you were initially seeing that the macrophages looked like they were clearing the bacteria. But then they were causing death through a mitochondrial mechanism that involved cyclophilin D. And the cell was undergoing a caspase independent death, a necrotic death. We also noted that this required RIPK1, RIPK3, and a mitochondrial phosphatase PGAM5. Okay, so the next question that Fran asked was, how does TNF through these three determinants induce mitochondrial reactive oxygen species or ROS? So I'm going to remind you about some basic biochemistry. So normally you get some reactive oxygen species formed when electrons flow through the electron transport chain. So remember that products of metabolism will give you NADH, and then as this hits complex 1, the electrons flow through the complexes, and as they flow through complex 1, some of the electrons will combine with will combine with molecular oxygen and will give you reactive oxygen species, and this is a well-known thing. And what we showed. Now, I should tell you one thing. There's an insecticide called rotenone, which inhibits complex 1. And if so, what it'll do is, because the electrons can't flow, you'll actually increase the amount of ROS that forms. And what we saw was that if you didn't have high TNF, infection gave you a little bit of increased ROS. That is, just the infection alone was giving you a little bit of ROS. But this was through forward electron transport, because if you treated with rotenone, it gave you even a further increase. Now, we had expected that with high TNF, all of this would just be going up even more, but we were surprised to find that actually we had the opposite result. You got way more ROS with TNF with infection in TNF. But if you treat it with rotenone, then it went all the way back to baseline. And it turned out that this is a well-known phenomenon. And what it is a phenomenon called reverse electron transfer, where electrons flow backwards from complex 2 into complex 1. And that's why the rotenone reduces this. And that is a signature feature of reverse electron transfer. Now, reverse electron transfer is known to occur when you have an increased pool of reduced CoQ-H2 with a high proton motive force across the mitochondrial inner membrane. And it was initially identified as a sort of an artifact in isolated mitochondria in, I believe, 1960. But lately, it's been shown to have pathological roles, for example, in strokes and heart attacks. And in fact, Mike Murphy is my colleague down the street in the mitochondrial biology unit, who's the one who showed this pathological role for reactive oxygen species. And it typically forms when succinate accumulates. Let me just take you back one. Because usually when when you have an excess over-oxidation of succinate is when you're going to get an over-reduction of CoQ. Okay, so we knew now that we had reverse electron transfer, that this most likely was due to an over-oxidation of succinate. And succinate is made in the Krebs cycle. And so the question is, so now we wanted to know what was the source of the succinate. And there's, as you know, if you go back to your elementary biochemistry, you can get different pathways feeding into the Krebs cycle. Glycolysis does, fatty acid oxidation does, and finally glutaminolysis can do this. And so we were able to target all of these pathways to try to figure out which one it was through using very specific inhibitors. And we realized that what was upregulated in the presence of high TNF was glutaminolysis. And specifically what was upregulated was glutamine transport into the cell. So when excess of glutamine came into the cell, there were very specific glutamine transporters. And we think that this is because they're most likely getting phosphorylated by RIP3, and this is why you're getting increased glutamine uptake, which leads to excess glutaminolysis because that's converted to glutamate, and then you get alpha-ketoglutarate going to succinate. Okay, now, all this was inferential through many very clever pharmacological and genetic manipulations that Francisco did. But of course, we wanted to see if there was biochemical proof. So we went to Mike Murphy, and he has very fancy state-of-the-art mass spec facilities. And remember, these fish are tiny, right? These embryos are tiny, but we were able to show that indeed you get high succinate with high TNF. And then this comes down if you inhibit either glutamine transport into the cell, or if you inhibit glutamine to glutamate conversion. And furthermore, we were able to also show that, remember, this is dependent on RIPK3 and PGAM5. And again, we were able to show that if you knocked out RIPK3 and PGAM5, you lost the succinate. So it was all looking very good. So to summarize just this part of the story, what you can see here is that when you have high TNF, and when normally in wild type infection, you make a little bit of ROS, the bacteria, but it doesn't kill the bacteria, and they grow to some extent in a cellular granuloma. But when you have high TNF, then the combination of the bacteria and the high TNF cause the granuloma to undergo necrosis. And this is because of reverse electron transfer that leads through succinate, that leads to an excess of ROS production, and that then kills the cell. And what was really cool was that we were able to identify a well-known human and safe human drug, metformin, that's widely used for diabetes as an inhibitor of complex 1. And at least in the fish, we were able to show that this can completely reverse the pathology and susceptibility of the high TNF fish. But of course, this now, yes, so we solved the question of high TNF, how high TNF makes mitochondrial ROS through this zany mechanism of reverse electron transfer. But now the question was, well, okay, but why does the excess ROS lead to macrophage necrosis? What's the mechanism for that? Fran also figured that out, and it turns out to be an elaborate pathway that flows through the mitochondrion of the macrophage, the lysosome, and then finally goes to the ER. So let me walk you through this. So we've already gone into how ROS is produced. What happens next is the ROS diffuses into the lysosome. I already alluded to this because at first it begins to kill the bacteria. But what happens at the same time is that it activates a lysosomal enzyme called acid sphingomyelinase. Acid sphingomyelinase produces a lipid called ceramide. That's its product. And ceramide activates a lysosomal cathepsin called cathepsin D. Cathepsin D comes out of the lysosome and activates a canonical cell death protein called BID, which activates a cell death protein called BAX. Now, BAX is the protein of apoptosis. It goes to the outer mitochondrial membrane and causes release of cytochrome C, and the cell dies through a caspase-dependent death. But in our case, we knew that our death was not caspase dependent, nor was BAX going and working at the mitochondrion. We could show all of this genetically. We being Fran. What Fran showed that instead what was happening was that BAX had a completely new function. It was going to the ER, and it was activating the ryanodine receptor, which is one of the two calcium channel transporters in the cell. And so it was causing calcium to flow from the ER into the mitochondrion through the mitochondrial calcium uniporter. And this calcium was triggering cyclophilin D, which is a mitochondrial matrix protein, to translocate to the inner mitochondrial membrane to cause the opening of the mitochondrial transition pore complex that cause or dysregulation of the complex. And this then causes loss of voltage potential, and the cell dies in necrotic death. Now, this pathway was cool, but Fran was able to show that there were lots of druggable targets. So I already showed you right from the first part where we could see that ROS was made through complex 1, that metformin, which inhibits complex 1, could block it. So we already had one targeting drug. But then if you went down the pathway, you could find more. Commonly used antidepressants, tricyclic antidepressants like desipramine, inhibit acid sphingomyelinase, and we were able to show that this too would inhibit the pathway. You could show that inhibiting ryanodine receptor with a drug called dantrolene could now cause loss of calcium flow, and that would inhibit the pathway. But in a way, the cleverest idea that Fran had was, wait a minute, ultimately, this whole pathway is because of calcium overload in the mitochondrion, going from the ER, which is the normal repository of calcium, into the mitochondrion. And he said, what if I just reduce calcium type calcium channels that are on the plasma membrane, which macrophages have. And in fact, there are well-known drugs that do this. Some of the most commonly used drugs do this: verapamil, nifedipine, diltiazem. And when Fran treated the fish with these, lo and behold, he was able to show that, yes, if he just reduced calcium flow into the cell, the whole pathway was now stopped, and the TNF high fish or the LTA4H high fish were no longer susceptible. And of these, one of the most interesting to us, they're all very interesting drugs. And by the way, these are all well-known human drugs, pennies to a pill. So, very good to try as adjunctive treatments for TB. One drug that we're particularly interested in is verapamil, because we'd already landed on verapamil before through a completely different mechanism. So when Kristen Adams was a graduate student in my lab many years ago, she showed that a completely different project. She showed that when mycobacteria enter macrophages, they become tolerant to multiple antibiotics. And by that, I mean, they don't become genetically resistant. It's just that they don't get killed by the antibiotic, even though they're not genetically resistant. And she was able to show that this was because these bacteria expressed bacterial efflux pumps that kick out the antibiotic. And they do that only when they get into macrophages. So these efflux pumps are induced when they enter macrophages. These kick out the antibiotics, and so they become tolerant. And she was able to show that multiple drugs, human drugs that are known if bacterial efflux pump inhibitors, inhibit this tolerance and make the bacteria more likely to be killed by the antibiotic. And her favorite drug from that list was none other than verapamil. So you could see that you might get a twofer with verapamil because you would be able to inhibit drug tolerance as well as this new mechanism of inhibiting high TNF necrosis that Fan has identified. Okay, so that brings me to the end of my talk. I hope that I've convinced you that because TB is an ancient disease, Mycobacteria have had a lot of experience dealing with us. And so, of course, they're exquisite immunologists, cell biologists, biochemists. You know, they've known that they've figured out long before we even knew about BACs that BACs can interact with the ryanodine receptors in the ER. They've figured out reverse electron transfer way before Britton Chance did, and so on. And so because of this, studying TB can uncover new biology, which is, I hope I've convinced you of. You know, for example, we've shown a new role for reverse electron transfer. We've shown a completely new function for BACs, and cathepsin D, which is normally a lysosomal enzyme that has very little lysosomal function. So when people knock out cathepsin D in mice, their lysosomes are okay, and yet these mice do terribly. And therefore, cathepsin D has some extra lysosomal function, and it appears that one of those might be to cause activation of BID, and that then leads to this function. So you can see that you can do a lot of interesting biology by using TB as a probe for it. But on the disease side, we've identified multiple host targeting drugs that are well-known drugs. And because these drugs target the host and therefore thwart the bug's pathways that it uses for pathogenesis, it should be useful for both drug-sensitive and drug-resistant TB. And finally, I should point out that what we've shown you here is an ideal example of precision medicine, namely genotype-guided therapies, because I've shown you that these drugs will work in the high TNF state, but not in the low TNF state, which is, you know, very much where, for example, cancer therapies are going. So, of course, any good project or a series of good projects always leads to more questions, and many questions are of great interest to us. And if they are to you, you know, do contact me. But for example, one question is, how does ROS get across, and how does calcium get across? There's got to be organellar contacts, and organellar contacts are becoming quite prominent in the field of in a homeostatic context. They're supposed to regulate many cellular functions, and we suspect that they're having a pathogenic role here, and it would be fun to be thinking along those lines. And actually, someone in my lab, Mikhail Eisenberg, has come from Israel, from a contact site lab to look at this very question. We'd be interested to know how cathepsin D does its business. How does it get out of the cell? How does BACs engage with the ryanodine receptor? This is all completely new. How does TNF activate RIPK1? What is the role of PGAM5? You know, that causes the ROS to be produced. How do the glutamine transporters get phosphorylated, and on and on. And so with that, I'll close and just remind you that we mustn't forget that fish are friends, and I hope that you'll agree with me that we've learned an awful lot from them. And with that, I thank you for your attention.
Great. Thank you so much, Lali. That was a beautiful talk. Such a beautiful example of integration also of, you know, mechanism, model organisms, new model organisms in terms of thinking about ways of studying pathogenesis and infection, and then human, direct connection to human genetics and relevance. So thank you so much for that beautiful talk. As we always encourage our listeners, and not only the people who are listening today, but this video will be played on the YouTube channel for the foreseeable future, forever. And so there's always a chance to contact Lali. Fozia, could you please show that slide again real fast? The one with the Twitter. There we go. Yeah, so if you can go to on the Twitter account of Global IMMUNOTALKS, you'll find a question that says ask questions for Dr. Ramakrishnan here. And then go ahead and type in your question. And if you want to put hashtag Global Immuno, that other people can find it easily as well. And so again, thank you so much, Lali, for being our speaker and hosting us with a beautiful hour of science. And I hope to see you again soon. So
likewise, thank you very much. And thank you for doing this. I've been enjoying the talks, and so I really appreciate the service you're doing for the community, you and your colleagues.
Yeah, and again, thanks Alina and Carla for initiating this. This is a great program. So thank you.
Bye-bye.
Bye.